All right, we're going to go ahead and get started. First of all, can people hear me?
Yes.
All righty, we'll get started then. Well, good morning for those of you who are joining us on the West Coast, and good afternoon for those who are joining us on the East. Welcome to the Brain Image Library's Data Submission Workshop. Before we get started, what I would like to do is introduce the team to you. Each team member can introduce themselves in more details whenever they are going to give their talk today. So my name is Alex Ropoleski. I'm the lead PI for the Brain Image Library. Joining me as well is Alan Watson from the University of Pittsburgh as one of our MPIs on the project. He's with us here today. Also with us here today, you're going to be hearing from our data curator, Mariah Kenny. She will be going over metadata and answering all your questions on that. We're going to have Luke Tudi give a presentation on how to submit data to the Brain Image Library. And we're going to have Ivan give a brief introduction to some of the computational resources that we have here at the Brain Image Library. So if I can get my slide to advance, we'll be in good shape here. There we go. So that's essentially what the workshop agenda is for today. When we end, we may end a little bit after 4, but we'll stick around after 4 and answer any final questions that you may have for us. Likewise, for those of you who are joining us tomorrow, what we're going to do is, for those of you who are joining us tomorrow, you can join slightly later tomorrow because there are a few new people who will be joining us just for tomorrow's agenda. And what we're going to start off with is a basic setup to ensure that people's accounts are working okay, so that there is no interruptions during the hands-on session, and we have account issues straightened out if there are any before we get to the hands-on session. I will then review the content from day 1, just a very brief introduction, and also give participants the opportunity to ask any questions that they may still have from the day 1 content. Then Mariah, we'll turn it over to Mariah, who will go over how to find data in the Brain Image Library archive. Luke is going to give a demonstration of a new tool that we have available called Open On Demand. And Open On Demand will allow people to submit jobs to our cluster through a web interface, as well as allow people to explore using Jupyter Notebooks. We're then going to turn things over to Iyana, who I just realized I forgot to introduce her earlier on the earlier slide, but she is joining us as well. And she's going to work through a Jupyter Notebook exercise with everyone on image registration. And then following Iyana's talk and exercise, we're going to turn it back over to Yvonne, who will tell us a bit more about how to run different packages that we have here at the Brain Image Library. So with that said, just a few logistics as far as the workshop's concerned. First of all, the lecture notes will be posted on the Slack workspace. Everyone should have received an invite to the Slack workspace. If you did not, please use the reactions function of Zoom and raise your hand, and one of us will, or just simply post a message in the chat, and one of the team members will take care of that issue and send you an invite so that you can use the Slack workspace. Also, these lecture notes that I'm using right now are posted in the Slack workspace under the day one, the data submission workshop Slack channel. Please use the Slack channel if you have any questions that you ask. I mean, you can use the chat as well, but if you use the Slack channel, those will be kept around longer so that other people can peruse those questions if they have the same question. And we will review all the questions that come in through the end of the week and respond to them. If you have questions after the week is done, what we would like you to do is to just send email to our hotline, and our hotline email address is bill-support@psc.edu. Everyone who has signed up for the workshop should have received a username in the email. If you already had an existing account at Bill, you may not have received that username and email, but you should use the username that you are already using with Bill. If you do not know what your username is and did not receive that email, again, just post in the chat right now, and one of the team members will find out what your username is. So once you've received your username, there were instructions in that email on how to set your initial password. If it's been a while that you've used the Bill system and you don't remember your password and there's an issue, you can reset your password at the address apr.psc.edu. If you have an existing account, there's no need to reset your password at this time, but if you don't remember it, that's how you would go about doing it. So now we're going to briefly go through an exercise to have everyone check their accounts and make sure that they can log in properly. And what we are going to use for this exercise is a piece of software called X2Go. And that will allow you to log into a machine at the Brain Image Library. And once you log into that machine, if you can give us a thumbs up in the reactions, the Zoom reactions, that would be great, so we know when to move on to the next section. So briefly, what I'm going to do is the next slide has some brief instructions on, if it will advance for me. There we go. Have some brief instructions on how to use X2Go. So if you have not installed X2Go, please do so at this point in time. So the instructions on using X2Go is you will start X2Go on your computer, and then under the session menu, you're going to select New Session. And then in the Session Preferences menu, it's a pop-up menu, you're going to want to make several changes. You're going to want to set the session name to workshop.brainimagelibrary.org. And under server, you're going to want to set the host to workshop.brainimagelibrary.org as well. Under the session type, you're going to want to choose the selection Mate or Mate, M-A-T-E. And then you're going to click the OK button. Once you do that, you're going to then move the mouse cursor to the light gray box over the words workshop.brainimagelibrary.org and click on the left mouse button. Then enter your login name. This is your Bill username, and then your password, and then click the OK button. And after a few seconds, an X2Go desktop will appear on your screen. Again, once you've worked through this exercise, when it's complete, please go to the Zoom reactions and give a thumbs up. So I'm going to leave this up for just a few more seconds here. And what I'm going to ask Luke to do next is to give a demonstration on connecting using X2Go.
Just let me know when I should steal the screen from you.
My app won't open because it's unverified. I know there's a way. I'm on Mac. Does anybody remember? I've done it before, how to open an unverified app.
I'm not a Mac person. Yvonne or Mariah,
Yes.
if you know the answer, can you?
Yes, you have to go to the system preferences and then to security. And if you just try to open, let me see if I can, I'm opening my, and then there's like a security and privacy.
Actually, you know, if you have that ready, do you just want to give the X to go, you know, share the screen and demonstrate that?
Myself, I know mine it's already verified, so I won't be able to replicate.
Yeah.
But I can show you. I can show you share my screen if you want for 1 second.
Sure.
Let me share. So go to system preferences.
Yeah.
And there's one called security or privacy. Let me try to figure out what it is. Okay, here you go. Security and privacy. And if you just try open it, to just open X2Go and you got that message, this part here of your screen will look a little bit different, and it will ask you whether you want to open it unverified.
Oh, yeah.
And then just do that, and then it will. You just have to say yes, and it will let you open it.
Perfect. Thank you.
No problem.
Great. Luke, do you want to go through the demonstration?
Sure thing. So like Alex had mentioned, these are the steps for creating that session. So when you have this screen up, you'll go up to the top left here and there's an icon. This is for creating a new session. You can name this whatever you want. For the sake of the workshop, I'm just going to name it 2022 Somerville Workshop. The host will be the workshop.brainimagelibrary.org. And then down here is where you'll select that. This will let me scroll here. Give me 1 moment. There's the mate right there. Sorry, it was a little covered up. So doing that and pressing OK will give you a session on the right side of your screen here. If you double click, you'll get the prompt to log in. So this will be the login that you set up for the Brain Image Library. Okay, there will pop up with a session. And I'm gonna have to share my whole screen here so you guys can see all of this. So it's popped up with my session for a desktop. You have to log in here as well. And you should have access to your desktop. I can put up those settings preferences on the screen here so folks can see that as well.
So if you have been able to log in, if you can use the Zoom reactions and give us a thumbs up so that we know to move on. Likewise, if you need some help, feel free to post a note in the chat and one of the team members will give you a hand.
you started a new session, but don't see where to log in. Are you talking about on this screen or after you've typed in your login to get into the desktop?
I think I filled out everything the way you did there, and then I clicked, what is it, continue or okay, and then it closed that, and now I have the blue screen.
Yeah, so over on the right side of your screen, that should be over here now after you.
Uh-huh.
And then if you double click that, it will prompt you for a login.
Ah, okay, got it,
Sure.
thanks.
Yeah, and there was another question too. Sometimes whenever X2Go starts up the terminal screen, it's actually behind your X2Go session, so you may have to move windows around to find the remote system.
Double clicking that does not prompt a login for me.
Yeah, I can't, it will not open up the login.
But you do have it on the right side of your screen.
Yes.
What system are you using?
Mac 10.13.6.
Just drag, it's not, never mind. I thought that was an option too.
Should I just get rid of my current one and redo it?
That might, yeah, perhaps that might do something different.
I'll try that.
I'll leave these preferences up for you just in case maybe
Yes.
something got changed there.
Is anyone else still having issues? Everyone else is good to go on? If you're not, please speak up now or post a note in the chat.
For some operating systems, yes, X2Go requires Xports to launch that desktop screen.
I still can't get it to pull up login credentials. I recreated it.
Okay, what I'm going to ask Luke to do is to go into a session with you,
Okay.
and then you guys can work that out.
Okay.
Who am I speaking with?
Chris.
Chris. Okay, yeah, Chris. Let's hop over to a breakout, and then we'll fix that for you.
Okay. Thanks.
Does anybody else need to go into a breakout with one of my team members to work out an issue?
Yes, please. I'm dealing with the export.
Okay.
Okay.
This
you
is
wanna-
Asia.
Do you want to have them go in the same room?
Yeah, that's
And then
fine.
you and Yvonne can kind of team up with whoever needs help.
Sure thing.
Thanks.
All right, sounds good. I'll share my screen again and oh, Okay. For whatever reason, my PowerPoint is going crazy here. All right, guys. So we worked through the X2Go. Those who haven't worked through the X2Go are going to join a breakout room with Yvonne and Luke and work out issues that that you guys may have. So I'm just going to move on then and talk a little bit about the Brain Image Library and what the Brain Image Library is all about. So the Brain Image Library, what we are is we're a national public resource enabling researchers to meet data sharing requirements of the National Institutes of Health and the NIH Brain Initiative. The way that we enable people to do that is through our scope. And what we are is we are a permanent repository for high quality brain microscopy data sets. So if you're producing a microscopy related data set or microscopy produced data set, and you have data that is of interest to the Brain Initiative, that is if you are Brain Initiative funded or if you have some other research interest, but the data that you're producing might be useful for the Brain Initiative, you're welcome to deposit it in the Brain Image Library. There's no charge for depositing data. There's no limits to the size of the data that needs to be deposited, but we do request that if you are depositing more than 50 terabytes in a year, to please contact us ahead of time and discuss your data requirements with us. So in addition to providing a place for people to put their data, we also provide an analysis ecosystem with desktop visualization as well as high performance computing capability to enable people to process their data before they submit it, as well as use the data that other people have submitted to the library. And there is no charge for any of our services. A unique service that we do offer through our library as well is we provide support in not only through a hotline, but also, with large data sets, a lot of times there's issues with the local network in transferring data. And we do have networking expertise on staff who can be in touch with you to help resolve any network issues that are related to data transfer. So the scope of our data, what we typically receive is whole and partial brain image data sets of the mouse, human, other primates, model organisms, along with the higher level aligned and tracing data. We also receive data that is targeted experimental data, including connectivity between cells and experiments that do that, as well as spatial transcriptomics data as well. We also, as part of our mission, accept data that people may have acquired over the course of their career. There are some collections out there, some very good microscopy collections that are out there that people need a place to house on a more permanent basis. Say, for example, somebody is approaching retirement and they still want to make the data. They've been maintaining the data and making it available through their local resources, but now they need to find somebody else willing to take over the maintenance of that collection. we would be happy to discuss that with you as well. So in terms of deposited data coverage for what we have existing in Bill right now, the majority of our data is whole brain image data from mice. We do have a smaller collection of data from other species, such as marmosets, macaques, and we do have some human data, but it is not, it's not whole brain data. It's mostly from patch-seq experiments. In terms of technology, we have a lot of tracing data, but we also have some very high resolution full brain volumetric data, including data that has been achieved through the FMOS technology or the single two photon technology. We do have some light sheet data as well. And we do have some spatial transcriptomic data. So if people have spatial transcriptomic data or FISH data that they want to contribute to the library, we do have growing collections of those technologies as well. I'm now briefly going to go over the Bill analysis ecosystem and talk about some of the resources that are in that ecosystem that we will be using for this workshop. I do once again want to note that the Bill resources are available for you to use to explore Bill data in place, as well as for you to process data that you want to contribute to Bill. And these resources are available at no charge. Another point that I wanted to mention is that if there's data sets that you want to use and you want to incorporate some of the data in a class that you're teaching or a workshop that you're teaching, we do permit that, that use as well. And if you are teaching a course and or a workshop and you need resources for a particular time window, please let us know and we can arrange to give you unique access and exclusive access to a subset of our resources, again, at no charge to you. I'm now briefly going to go over the different boxes that make a part of the analysis ecosystem. The first system that I want to talk about is our storage system. Our storage system is a multi-petabyte system, and this is where most of the data will be stored. So our public data is on the Bill file system. It's down a special directory called /bill/data, and so everything that's public can be found down that tree. If you're uploading data, that's going to be somewhere, it's going to be on Bill as well, but it's going to be on /bill/lz and then a unique directory that only you have access to. Sometimes what we find is that people want to collaborate on a project, and we can make project space available as well on the Bill file system. And that project space is down /bill/proj. If you need to set up project space, please let us know through our hotline and we can set up that project space for you. You also have a home directory that's on the Bill file system down /bill/users, and you can feel free to use space there as well. I do want to mention that all of the systems that surround this Bill file system on the diagram here, they all have access to the Bill storage system. So whether you're using our computational cluster, which we're going to be talking about in a little while, or some of our high performance computing resources such as the Bridges 2 system or the NeoCortex system, all of those systems have direct access to the Bill file systems, and they all have high speed access to to Bill. So what we're principally going to be using in this workshop and the workshop tomorrow for computation is the Bill computational cluster. The Bill computational cluster consists of 8 large memory nodes. Each of those nodes has 3 terabytes of RAM and 7 terabytes of local fast NVMe scratch space. In the near future, we've received a machine that's in the process of being set up. That is going to be a GPU node that will be added to the Bill computational cluster. That GPU node will have 8 A100 80 gigabyte GPUs. Those GPUs will be connected with NVLink, so multiple GPUs can be used at a single time. And we are excited to make that resource available. We expect it to be up and running sometime within the next month or two. The main way to transfer data both to the Bill system and download data from the Bill system are through our Bill data transfer nodes. We have 2 data transfer nodes. They will be talked about in the workshop, particularly the upload transfer node at upload.brainimagelibrary.org and download.brainimagelibrary.org. If you would open a web browser and go to download.brainimagelibrary.org, you will see all the files that have been deposited in Bill that are public, made available in Bill data. A unique part of the Bill analysis ecosystem is we have a number of virtual machines that we do make available for and do serve different purposes. These are some of the virtual machines that you'll be interacting with for this workshop. We already with the X2Go exercise, we connected to workshop.brainimagelibrary.org. And that through the X2Go software and that virtual machine, you can do remote desktop applications directly from that VM. Likewise, we have another virtual machine that's set up called login.brainimagelibrary.org. And this is a convenient node VM that you can connect to using any secure shell protocol and submit parallel jobs to our Bill computational cluster, check on things that may be on the Bill file system. It essentially gives you Unix shell access to our system. Likewise, we can also set up virtual machines for specific purposes or specific users. Let's say that you're developing an application that will use the Bill data. It might be a web serving application. It may be it may be some commercial software that you have a license to that you want to use and use it to access some of the data on Bill. We can set up a virtual machine for you to enable that use case. The data submission portal, this is something that Luke will be talking about quite extensively in this workshop today. It's available at submit.brainimagelibrary.org. We also have a DOI portal that serves our DOI landing pages at doi.brainimagelibrary.org. And Mariah will talk a little bit about that tomorrow. And we also have a VM called ondemand.brainimagelibrary.org that serves as our on-demand web portal. Again, we'll be talking about that principally tomorrow as well. And the on-demand portal enables users to both submit jobs to the Bill cluster through a web interface, and it also enables users to use Jupyter notebooks. some systems that are part of the bill analysis system that we're not going to be using in this workshop, but I do want to make you aware of, is our bill tape system. And we do have the capability at Bill to receive data on LTO tape. And again, if you have a lot of data that you need to transfer to us, this may be a way to do that. We encourage you to contact us at billsupport@psc.edu if you want to explore this option, or perhaps explore our network analysis service to see if we can resolve some network issues that you may have in transferring your data. Likewise, what's not in the scope of this workshop is using our Bridges to High Performance Computing system or our NeoCortex system. But you can get access to both of those, and I do have some URLs listed on this slide that you can go to if you want to find out some more information about accessing those systems. Again, if you have any questions, you can email us at bill-support@psc.edu, and we will be happy to guide you however and answer any questions that you may have. As the computational resources that we make available through Bill, there's a number of software packages that are available for you to use. I do want to also make the point that we are willing to install what you need as long as there's no licensing fees involved to make you be more productive. And this list here is just a subset of some of the software that we make available through our analysis ecosystem that you can use. I did want to give people here a little note towards the future. A lot of our effort over the next few years is going to be in the area of making our data more accessible. And part of making our data more accessible is we're developing a way to serve data in the format that is needed for the application. For example, what we would like to be able to do is to allow people to store the data once and perhaps serve that data in, let's say, somebody's using the NeuroGlancer application to serve the bill data in a way, in the format that the NeuroGlancer program understands. Likewise, there's a lot of these other different programs out there that require the data to be in TIFF format, for example. So we could serve the data in TIFF format, in NeuroGlancer format, in whatever format the application needs. Part of this effort will also make a instance available directly from the Bill VM system that will allow users to explore data visually in place at Bill. So with that, I would like to thank all the data contributors to the Brain Image Library, and thank my whole team for all their hard work in making the Brain Image Library available and a resource that is useful for the neuroscience community. So with that, I'd be happy to take a quick question break if there's a question or two. If not, I will turn it over to Mariah, who will go over the metadata schema that is in use by the Brain Image Library. I apologize if I pronounce this name wrong. Is it Adijo? You have your hand up.
Yes, thank you for the, my name is Adejoke Elizabeth. Thank you for the wonderful presentation. Can you hear me, sir? Can you hear me?
Yes, yes.
Okay, talking about, I saw one of the presentation showed a graph where histology slide is underrepresented. So can I send data set from published work? Will it be acceptable?
Could you send it from a public?
Published work. I have a
Oh,
data
yes,
set from
absolutely.
BrainSlide. I've published it, but on histology of in the neocortex, hippocampus, but is H&E, Christopher stain, and immunostain. But it has been published. Can I still send it?
Yes, as long as you are the creator of that and you have the rights to those images, yes, you can deposit those in Bill, and we can
Okay,
issue a DOI for those as well. And typically, since this is already published, we can make it available. We can assign a DOI, but if you have when we assign the DOI, if you have the publication that it's in and you give us that reference, we will make that
Okay.
part of the metadata entry as well.
Okay, so if the second question, please, when submitting this image data, how will it be labeled? Because each photomicrograph represents different reports. So will it be attached with a document like a figure legend for the image sent?
So typically what we have is whole brain slices. So I guess it really depends on these H&E slices. Are they part of a what you would consider a single volume brain, or are they from many different brains?
A single volume brain.
Single volume brain.
Yes.
Are they ordered? Can you put them in a sequential order, or are they just more of a random order?
Okay. Oh, it can be put in it's not random order. It can be random order. But we have some data sets that are also in groups, like we have different groups and different treatments.
Okay. Well,
Yeah.
what I would suggest, so Mariah has there's a couple different scenarios in the way people deposit data. What I would
Okay.
suggest is that we move on to Mariah's presentation, and if it doesn't become clear after that presentation, feel free to ask your question again, or contact us through the hotline, and we can work through the proper way to submit your data.
All right. Thank you very much.
You're welcome. Any other questions? All right, Mariah, do you want to share your screen?
Sure. Can you all see it?
Yes.
Yes. Okay, perfect. So first off, I'll put my camera on so you can see me a little bit here. Hi, my name is Mariah Kenny. I am the data curator here at the Brain Image Library. And today what I'm going to talk to you about is the metadata, how to share your data, what's the metadata that needs to go with it, in a way that not only makes this easy for you to share your data, but also for the scientific community to be able to access it, reuse it, and have the information that they need to understand your data completely. So let's look at a snapshot of the submission process. So ideally, we've gotten through these first two boxes, which are creating your account, requesting access to the submission portal. But what I'm going to talk about today is really this box right here, uploading your data via the portal, what information needs to go in there, and what are the pieces of information that you need to be thinking about whenever you're uploading your data and metadata. These two happen simultaneously because the organization really of your data impacts your metadata. So it's important to think about these things right after you've created your submission. The rest of these steps are what Luke is going to present on afterwards. So don't be worried if it's a little unclear right now of the steps that you actually need to do in the portal. So let's talk a little bit about what are the goals of the bill metadata and why is it designed the way that it is. Really, what we're interested in here is, as Alex mentioned, is to make sure that our data is accessible and to increase the fairness or the findability, accessibility, interoperability, and reuse of the data, so that we're ensuring that the data that is generated is useful to the scientific community. One large part of making sure that it's findable and accessible is making sure that there are DOIs and ways to access and cite the data that is deposited in bill. So the metadata schema is definitely designed with the ability to issue DOIs in mind. So DOIs currently are not issued automatically, but in the near future, that is the way that we're going. I'll talk a little bit more about DOIs at the end, so you can see exactly what that looks like, what that means, and how DOIs are issued. But also, bills, the metadata is practical because it is implemented in a spreadsheet way. There's no spreadsheets are easy for us to put in data and to organize all of our data. I'm sure most of your metadata for your samples is already in a spreadsheet format, so it's easily compatible there. You can do some copying and pasting if you need to. But also we have these metadata models that I'll be talking about that allow us to remove redundant fields in your metadata, so that copying and pasting can also lead to errors, right? So we can reduce that by making sure that we get rid of any redundant fields. If you're interested in reading more about the metadata schema and the ideas that went into it in order to create it, you can find that information at this preprint on archive. So when I talk about metadata, what does that mean? What is the information that we're actually asking for? We'll go through an example in a bit about exactly how to put in the metadata and what the exact fields that are being asked for. But as an overview, there are 7 tabs within the spreadsheet, one being the contributors, who's getting credit for this data, who should be contacted with questions and things like that. Not only can you give credit to specific individuals, but also institutional groups, funding information, who provide financial support for the data collection, publication information, if this data is associated with an already published data set or a preprint, or there's any protocols that can also be associated with this data. All of that can go into the metadata and we can link those. Then, so the first 3 are pretty much how to find and access the data, but the last 4 are really how to reuse the data, describing the data in depth. What instrument was the data, where your data files captured with, data set information, descriptions, and also where is it at whenever you've uploaded it, specimen information, what organ are they from, samples, donor information, and also the image metadata as well, landmarks, channels, sizes, all of it. So as I go through this, I'm going to be using 2 words with one, data set and submission. These 2 are going to come up a lot. So I want to define them right as we get started here. A data set is what we define as a standalone entry, an image volume, an image set. Normally this is associated with a single object, but I recognize that we all have, that science is always evolving and changing. And so it's can also be in whatever your experimental unit is of how you've collected this data that has unique metadata associated with it. So for example, if we're talking about a single subject here, and that has one genotype, and then I have another image set of a different individual that has a different genotype, these would be two different data sets because they have unique metadata. So as I said, it's normally associated with a single donor or subject, or a single part of the brain whenever there are different parts of the brain in a single submission. There are typically many data sets that make up a single submission, and these data sets contain many image files that are assembled to form complex multi-dimensional volumes. A submission contains one or more, can have one, they can have many data sets with their metadata. We do this because many of you might be needing to report your data every single quarter. So if you are collecting data every single quarter and it's all related, it wouldn't just be one, you have a submission every quarter, right? And you want a way to kind of be able to bring all of these different pieces together. So your submissions inherit project information. Luke's going to explain more about how to set up your different projects in a little bit. And in general, these smaller submissions are generally recommended because we do validate the data. Everything that is published in BIL is validated and metadata curated. So each level also of your data should generally be uploaded in separate submissions. This means raw data submitted separately from the data that's aligned to a reference or something like that. We would consider these two separate submissions. And that's just to make sure that data can be ingested properly and we can associate data within the database correctly. So what are the two main things that I want you to take away from that? One is that there are many data sets that can make up a single submission, and two, that each data set represents a single donor or individual or experimental unit. So I hope that clarifies what I mean when I say data set. This term can mean a lot of different things to different people. So next, we'll talk about metadata models. So whenever you upload your metadata, the portal will look something like this, and it'll ask you questions. What does your data look like? And you'll have to select one of these different options. These are the different models. So you'll choose a metadata model whenever you upload your metadata. We'll go through those in detail, but these metadata models describe the structure of your data. How are your data sets related to each other? This way we can ensure that your data is properly ingested, indexed within the database, and we can describe all of the relationships between the data sets correctly. The metadata won't be uploaded if your metadata model selected and the structure of the metadata file isn't correct and they don't align. So it's important to kind of consider these whenever you're creating your metadata file and uploading it. So what do I mean by these metadata models? Let's get into it. So, and what do these actually mean? So for the first metadata model, each data set is generated from a single sample or a donor. So you can have many data sets, many different samples, but they're all collected on the same instrument. So something like whole brain imaging of multiple mice, something like that would be would fit under this model. So these lines are connecting the metadata here. So this one sample has one data set, also has another metadata piece of image. So these are all going to be different lines on different tabs of the metadata sheet. I'll show you an example of this in a minute. The next metadata model is a data set was generated from multiple samples or donors on the same instrument. So if you have a data set and there are many samples included in it, but these can't be parsed out into different files or something like that. So this is more common maybe with processed or higher level data, where the samples can't be separated amongst different files. So that would be what this model would be used for. So you have many samples in a single data set, one image set, and it's all included in the same instrument metadata. The third metadata model is that all data sets were generated from one specimen, but are maybe from unique regions of interest on the specimen. So we have one sample, but they have multiple data sets because there are multiple regions of interest. These are all result in different images, but they have the same instrument metadata. And the last option here is that all data sets were generated from one specimen, but they use different machinery parameters. So you have one sample, but they result in many different data sets that are associated with an image that came from a different machinery or instrument metadata. So with that, let's look at an example real fast. So my example here is that I have 8 image files from 4 different samples or 4 different mice looking at the same part of the brain or even a full brain using the same microscopes imaging settings. So of these different models, I would say that metadata model 1 best describes this example. So let's look at how I would think about how am I going to organize my data files and how would I put that into my metadata, or how would I organize my data files so that I could easily put them into my metadata file. If I have 8 different images, these are this brain 1 and brain 2, this is an image set for me. And so my directory structure, these would all be in different folders. So each mouse here gets its own folder in the directory of where my submission data goes. So let's look at an example of this real fast. Let me pull up a metadata sheet and we will go through and look at how this metadata would be put in. So whenever you download the metadata sheet, it will look something like this. Let me make this bigger. Hold on one second. So the first thing that you'll have is the README file. Oh, I should mention that all of these files, if you want to look at this file with me, you can go to this Google Doc that was sent out earlier and is in your email. If you want to look at the examples, you can click right here and it'll take you to a folder that has all the metadata examples in it. Metadata example model 1 is what we're going to be looking at right now. So if you want to follow along with me, or you can do that. So let's look at the README here. This just describes some of the different notes and instructions for how to fill out your metadata file. There are things that are required fields or also controlled values, but we'll go through those in a moment here. If we look at contributors, it's the first tab. So you can have one or many contributors, just their first and last name or name of the organization. Creator, it says that the main creator of the data set, you can kind of think of this almost as a first author of the data set, or the contact person even. There is contributor type. So this is a controlled value. You select one of these options that best describes the role of the person who's listed in this row. Then there is name type. Is this a person or is this an organization? ORCID ID for that person. So we do require that the creator has to have an ORCID ID. Then this is ORCID saying that well, you can have more than one type of ID here. You could have an ORCID or a ROR, ROR for a different organization. There are a little list of the acceptable identifiers in this field. The most popular is definitely ORCID for individuals, though. Then the affiliation, where is this person affiliated with, or where were they affiliated with whenever the data was collected? And the identifier for that organization. So this one is for the University of Pittsburgh. This is the ROR identifier that I would use. And then what type of identifier? If we look at the second tab, this is the funder's information. So things like where did the financial support come from, the institution, again, the ROR identifier and what the type of identifier that is, the award number and the award title. Publication information. This doesn't have to just be a associated article. It can be different things like published, anything that's published and has a unique identifier, or persistent unique identifier, such as a DOI or PubMed ID or preprint. So you can put any type of DOI, even if you have things like if you publish your code and you want to put it in a place like Zenodo or something, and you have a DOI associated with that, you'll be able to put that DOI here. So those first three tabs, the format of those is the same for all of our different models here. But with model 1, let's take a look at it again, is that they're the 4 different samples from the same part of the brain using the same microscope settings. So we have one instrument row here. You can put the red is what is required, but I do encourage you to include as much information as possible so that the users who end up using your data are well informed of what your data is. I always try to get people to think about, if you were looking at your data for the first time, what is the information that you would need in order to reuse it? And that's the level of documentation that we really strive for here. So I have one row and that's instrument tab. And for my data set tab, I have 4 different rows. These are my 4 different data sets for my 4 different samples here. 4 different mice, they have 4 different data sets. Even though I have 8 images, every data set has its own subdirectory. Then with that, I have a short title for the data set. This is important because all data sets are issued DOIs, so a unique title for each data set. Social media, if you'd like, so that we can have some information to publicize your data if you'd like. It's optional though. And then this rights information, all data in Bill, especially if you're affiliated with the Brain Initiative, is CC BY 4.0. All data in Bill is under this license. If you have any questions about that, please feel free to reach out to me. Then things like general modality and technique, these are controlled values. So you can see here where it says CV, cell morphology, connectivity, population imaging, spatial transcriptomics, or other. If you see something here that, if there is nothing that really describes your data well, you can feel free to put other, and this information helps us so that we can constantly make sure that what we're doing is serving your data, and we're updating to make sure that you don't have to make things work. Rather, we want to be making sure that we're creating things so that your data is well represented. So general modality and technique, there are two different lists of controlled values here to best describe the technique used whenever your data was collected. Abstract is just a few sentences, ideally describing your data set. This information is also required for us to issue DOIs. So that's important. Then you can also put information about your methods or information about your other technical information that's needed in order to understand your data. So if you did something very specific to a few data sets, or process them in a different way that's not included in maybe the protocol that you included, that's where you could put this information. Then let's look at specimen. So I had the 4 different samples, right, of my 4 different mice. So I have 4 specimen lines here. This line 7, or row 7 in the specimen tab correlates to row 7 in the dataset tab. It's very important that these align. And so mouse 1 is probably a really bad local ID number for this example. Ideally, this is what's associated with the organism that you have. Maybe this is something that you, any identifier that you use whenever you are collecting your data. Species, normally mouse, human, any other model organism, NCBI taxonomy. These are all required fields. Age unit. So if I have a 10-week-old mouse, sex, need genotype information, a local ID of the organ. If there's a organ name or component of where the tissue is found. Sometimes people have a second ID for the samples. So if you've got a data set where you have one organism, but many samples and many images taken from that, that you can include here. There's an atlas used to describe the information in the location or the tissue, a region where the tissue was found. So I have 4 of those for each of my 4 different data sets. But now let's look at the last tab here, which is the image tab. It describes the data set's images, orientation, landmarks, channels, and sizes. So the required fields here are the x-axis, the predominant tissue direction. So all of these, I really encourage you all to, whenever you're filling these out, to take a good look at these description underneath the title of the fields here. They really do contain all of the information that you need to give you a good idea of what to put for your image data. So this one is the orientation of your x-axis. Then these are optional fields if your x-axis is oblique. There are some other controlled values for those. Y-axis, same for z-axis. Then the landmark, coordinates of the landmarks, channel numbers, display colors, things like that. I won't go through every single piece here, but just this is all of the information that you would need to understand what are the images, what are the parameters of these images, and what would I need in order to reuse these images. So with that, let's take a look at a second example. Example 2 being that the images are taken at 2 different magnifications from the same mouse. So if I was looking at these metadata models and getting ready to upload my metadata, I would say that the 4th metadata model is the most appropriate for it. All data sets were generated from one specimen, but using different experimental parameters or machinery parameters even. So if I have these 2 different magnifications, I have 2 different image sets from each magnification. In this example, I have 10x and 3x, 30x from the same mouse. What would that look like in a metadata sheet? We'll just take a brief look at this one. So contributors is the same as before. The funders, also the same as before. Publication, same as before. But let's look at the instrument tab. So in the last one, I just had 1 row on the instrument tab. And in this one, I have 2. Specifically, there's some unique metadata with these 2 fields, right? One being 30x and one being 10x for my 2 different data sets that I have. Again, this is the unique metadata here. So for each of those 2 different parameters, I have 2 different data sets that each lead to their own subdirectory and have all of the associated metadata for both of those. But both of these are the same mouse, right? So I only have 1 tab on the specimen metadata row. And then because these are 2 different image sets, I have 2 different rows on the image tab. So both of these examples are included in this that I shared with you. So you can take a look at these if you'd like. Oh. Now, we looked at that. Just to take a look at what an example would be from the 3rd option here, all data sets were generated from 1 specimen. So if I have 4 different image sets that have 4 different regions of interest from the same brain, that would be an example. I would have 4 different subdirectories for my submission for each of the different regions of interest. And then there's an example included that you can see exactly how that file would be set up. And this last one, this is metadata model 2. The data set was generated from multiple samples of specimens on the same instrument. So this one is I think the only option where there is only 1 data set included. So if we're talking about cases where there are multiple samples that each have unique metadata, but they can't be separated into their own files or directories, common with the higher level data, there would only be 1 data set here. They would have many rows in the sample column, but there's only 1 data set included. So let's take a look at what happens to this metadata afterwards. with this new metadata model and all of the information that you saw really does allow us to include fields that will allow us to issue DOIs automatically. This is a future development. Currently, DOIs are issued by request, but all data sets in the future will have a DOI associated with them. Let's take a look at one of these DOI pages looks like. So what will your data look like whenever your data does get published? You can see all this information, the metadata, all of that information that we talked about putting into the metadata sheet is described here. So, and then you can see funding information, contributors, and then where that data is on disk and in Bill. So this is at a single data set level. Again, because this is at a single data set level because data sets will have unique metadata associated with them. But and because many people do end up submitting data sets and metadata every single quarter that associated with a larger group of data. So in addition to data set DOIs, we also have group DOIs, which allow us to collect all of the different data sets that are associated with a single project and make them available in one place so that you can have one link if you would like to share in your publication or any type wherever you'd like to share your data. So specifically, let's look at this one. It has very similar metadata because that's what's required in order to issue a DOI, but I just wanted to call your attention to this related identifiers section at the bottom. These all have different collection numbers here. So these are all different data sets that were maybe uploaded at different times or uploaded in different submissions, but they're all available at the same place. So even if you do need to update or upload your metadata in different submissions, you can still, we can still work with you to make sure that your data is accessible at one place so that it's easy for you to share. And with that, I really do encourage you to reach out with questions. You can either reach out to Bill support here, my personal email is here as well. Whenever you are filling out your metadata files and you have questions or concerns, please feel free to reach out to me. That really is why I'm here. And if you have metadata that you don't feel the models accurately reflect, we can work together, and I'd be very glad to kind of come up with a custom solution for you to make sure that your data is well represented in the Brain Image Library. I know that science is constantly changing. The way that we collect data is constantly changing. And so we are definitely ready to adapt to whatever you need in order to share your data. So with that, if there are any questions, I'd be glad to answer them. If not,
I guess sorry, I do have one question. So I have a data set. I have like 50-something thousand neuron tracings.
Mhm.
And they're all gathered the same way and everything, but they have different levels of proofreading and things like that. So how would you record that in the metadata sheet?
Different levels of proofreading. I think I would need to maybe can I message you on Slack?
Yeah.
And we can chat about your data so that we can figure
For
out
sure.
how to get that in there, because that would be a lot of it's a lot of data. I want to make sure
Yeah.
that
Yeah. Yeah, maybe I should split it up too. Yeah, we should talk. Okay, thanks.
Yeah, no, totally. Yes, please. I'll message you and we'll talk about it.
Okay, thank you.
Awesome, thanks.
I have a question, if I can.
Sure.
I want to gauge if I understand correctly, like, I personally was gonna submit my data as a body of work in the form of a publication. So well, I guess, do people do that? It sounded like maybe they did. And I would envision the data sets being maybe data that contributes to a particular figure, and the whole submission being the data that is in the publication. Is that a fair way to do it?
So, I think I would have to know a little bit more about the specific type of data that you have here.
For example, a
yeah.
figure might consist of panels of confocal Z
Mhm.
projections, and then maybe the data you would want to share to submit would be the actual confocal stacks.
I see. So you would need to enter your data into one of these different models so that we could accurately have it indexed in the database. But maybe we can kind of include information on the DOI page that describes where each of the data is coming from and what figures and things like that it's described from in your publication. Not sure if that.
Yeah, so yeah, this is Alex. I could chime in a little bit, and I guess it really depends as well on what the requirements are from your funding agency.
Yeah.
I know with the Brain Initiative, if it's Brain Initiative funded, they
This
want
is
you
Brain
to
Initiative.
okay, so they want you to basically share all your data. So you would need to kind of submit the whole Z stack for that. If you want to reference a subset of that data set for a figure, I think we would need to discuss in a little bit more detail exactly what that would be and how we should index that appropriately.
Okay, it sounded like maybe model 1 would fit, but I'd probably have to talk to you for more information.
Yeah, model 1 would fit the Z stack.
Uh-huh.
I'm just not sure, exactly if you're going to subset things into, a figure, exactly how we would best represent that. I think we would need to, carry on this conversation with Mariah and understand a little bit better about what, figures can be lots of different things. So I think we would need to understand that a little bit more.
Sounds good. I'll reach out to you, Mariah. Thanks.
Good, thank you. Yeah. Yes, you have your hand raised, Ed, okay?
All right, thank you. Please. You mentioned about funding, Brain Initiative funding. Okay, if for me now, if I don't have funding from Brain Initiatives and I have some data, I can't send them. Can I still send them?
Yeah, so as long as your data is broadly related to the Brain Initiative and kind of has similar goals as the Brain Initiative, we can generally, yes, you would be able to publish your data in the Brain Image Library. You don't necessarily have to have the Brain Initiative funding, but it does need to be relevant.
All right. Thank you.
Yeah,
so
and
I
guess
read about the relevance.
Yeah, so
how
relevant.
Will I get to read about?
Yeah, I just want to say so what's relevant in my mind with whether it's relative to the Brain Initiative is we'll basically accept virtually any brain data set that's microscopy oriented. We won't accept a microscopy data set of, let's say, the heart, for example.
Yeah, I know that. Yeah, okay. So you can accept microscopy from confocal to light microscopy, stuff like that for histology. Yes. All right, thank you.
Any other questions before I hand it over to Luke? not, Luke can take it away.
All righty, let me get this set up. I think I'm sharing the correct screen here. Okay, can everybody see that?
No?
I can see your slides. Yeah.
Oh, you can see my slides, okay. Let me,
Yeah,
it's not in the presentation mode?
yes.
Yeah, you're good.
Oh, okay. Okay. Hi, everybody. My name is Luke Tewitt. I'm a web developer for the Brain Image Library project, but I also work closely with data contributors and users of the system as a whole. Today, I wanted to talk about the actual submission process for getting your data over to Bill. I'm going to talk about the workflow for depositing the data, how you would create a submission to do so, how to send the data over to us, and get all the necessary requirements in to get a public link for your data. We'll be providing metadata for the submissions. We won't be filling out one. We have some pre-made ones that Mariah had shown previously that we can use. And I also will talk about the PI dashboard that's available on the submission portal. So as Mariah showed, this is the overview of submitting data to Bill. Obviously, the first step is going to be creating an account and requesting access to Bill. I think the majority of you do have an account with Bill. If you don't, that's okay too. But that will start with creating an Xseed account. You'll visit the Xseed portal, fill out the account creation information there, and then you'll be given a username to Xseed, which you'll verify and then send over to us with via Bill support, our hotline, letting us know that you're a user who's interested in getting access to the submission portal. Generally, it takes a few hours to get that propagated to our systems, but once that happens, we let you know that you're good to go, and then you can go ahead and set your initial password, and that will all be finalized, and you'll have access to the submission portal. So it all starts with creating a submission on the submission portal. The submission portal has options to create a landing space for your data that you'll be submitting, and it gives access to a detailed view of your data submission information. On the website portal, there's an option under, and I'll show all of this in a interactive portion here, but there's an option on the top bar once you log in for a new submission, which prompts you to a form that asks for information about the data that you're submitting. So I'm going to go ahead and show that, and you can follow along as you please. So I'm going to go up to, if you're following along on X2Go, you can do the same thing, go up to Mozilla Firefox, and the submission portal is located at submit.brainimagelibrary.org. For you guys just starting, this will probably prompt you to log in. So once you do so, you'll be met with a screen like this. So we want to create a submission and a landing space for our data that we'll be submitting. So we'll go up to new submission. I'm just going to name this workshop data. For a description in the real case scenario, you'd want to be as descriptive as possible. Your organization name, your lab name, the project funder identifier. This will be the grant number associated with your data. I'm just going to put in some dummy information there just for the sake of time. And then down here, you'll have an option for project. If you've set up an account, you should have an option when you click on this for a project. If you don't, let us know. but you'll be started off with with a default project, which will just be your username and project one. If you, in the real scenario, if you're submitting for a grant or a or several grants, you'll have to let us know via the hotline, and just so we can get you set up to be able to submit to the correct places. But just for the sake of testing things out, you can always use your scratch space just to see how things go. So I'm going to hit save here. And now once I've done that, I now have a submission area to work with. And sorry. So now you created that submission. You have a few fields that get auto created for you that you'll need to take note of. Most notably the data path. The data path is where you'll be uploading that data to. It's an auto-generated identifier that's unique to your specific submission. And I can share that over here. If you go over to your view submissions, I'm going to have a lot more here than you guys do, but if you scroll down for the latest one we just did, this is our workshop. This is my workshop data submission that I just made. And this is the, if you scroll down here and see there's a data staging area. And this area is located under Bill LZ, which stands for landing zone. Your username, and then that number that's auto-generated, that will be a directory where you'll be uploading your data to. And as you can see, we haven't submitted any metadata or anything like that yet. It's just an area that's waiting for things to happen. So then we'll move on to actually submitting the data. So there's questions of how to structure your data. It's very, things are specific to whatever kind of data you're submitting or the experiment you've done. Generally speaking, the data submissions are broken up directory wise by each data set, or in some cases, if there's multiple experiments going on, an experiment and then data sets located under that. There's a lot more information of how to structure your data and good data practices on our website, which is linked there. It's under the brainimagellibrary.org, and then there's an option for instructions for good data practices. As Mariah mentioned, the data set, it's our definition as it's always changing in the science world is an object with a unique data piece associated with it. So an example would be a whole, a set of whole brains would be a data set, or a set of tracings could be a data set. So now we're going to move on to the actual data upload into that submission we created. So I have some test data available to you guys to run an rsync command on to move that data over into our submission area. You can use there's many methods you could use. You could use rsync, SFTP, Globus, SCP, but for the sake of this presentation, we're just going to use rsync. So I'm going to close out of this. If you go up to the top left here, there's an option for a terminal, and that will give you a command prompt. The data we're going to be moving is located at a directory called /bill workshops 2021. It's under data submission, data, and then images. So these are just two data sets of images that are pretty small, so we can move them over rather quickly. So if you take a look on your back on your submission portal, sorry, things running a little slow with everybody being on. let's make a note of that staging area we created. So my staging area, I can actually take an easier look at by going to /Bill LZ and then my name, my username being L296. So I have a lot of things here, but I can take a look specifically at the one I'm talking about, which is mine is 49CE. Or perhaps I didn't add that there. Oops. For folks following along, have you gotten to this point where you have a staging area, by the way? If I can get just thumbs up to see if we're on the same page. I don't want anybody to get too behind or anything like that. If you're having issues, please feel free to unmute and let me know. If not, we can go ahead with the data move. So I have several ones in here that are just empty that I can work with. So I'm going to work with this one here. In my terminal that I'm going to be moving this data over with, I'm going to type in rsync -ltr d capital D lowercase v-p. These are just flags that rsync sets. I'm going to point to that data on the Bill Workshops directory. And then I'm going to point to my landing zone directory. Which is this D7. So this will start moving data over into that directory. This is going to take a few minutes. So if there's any questions so far, please let me know, or any concerns or comments.
Will your slides be available for?
Yes.
Okay.
So while that's running, we can go back over into the presentation. Over to our uploading our metadata sheet. So you can do these things in tandem. You don't need to have the data there to upload your metadata. You can do that first if you like. You can do them at the same time. So we have on the same /Bill LZ /Bill Workshops, there is a folder called metadata. And this has a file in it that's pre-made for you to upload. If we go back to the submission portal and we go to new and then add metadata, we can add that metadata to our submission. The example I've given is the first option. It's each data set was generated from a single sample donor, etc. If we scroll down, we get a drop bar for associated collections, or associated submissions rather. And I'm going to attach that to the one I just created. If you want to download a new spreadsheet for yourself, they are available right here under the upload metadata form. You can download for LibreOffice or Excel. So I'm going to hit upload metadata there, and I'm going to click on that metadata sheet that I've pre-made. And if I go back over to my submissions, this gives an overview of all the metadata you've submitted for your project. If I go back to my submissions and then to my submission I just created, I can see that information's been properly set into my directory.
Question, Luke.
Sure.
Would the landing zone be created now, do you think? Now
It
that you've
should be created now unless there's an issue with the VM at the moment.
Yeah.
I noticed that it may not have been created for me. I didn't know if that was the same case for other folks.
Well, what I've noticed before is unless I upload the metadata, the landing zone is either not created or it seems like it's created but without right permissions. Or maybe it just takes a while. I'm not sure, but I've had to upload the metadata first. So that's one thing. But the other little bit more general issue is that in the metadata there is a cell for the landing zone or R24 directory. But then of course I need to create the submission for there to be a landing zone and R24 directory.
Right.
Is there an easier way to do it than creating a submission, then finding the R24 directory, then going back to the metadata and adding it and then uploading the metadata?
At the moment, that's the process. When you create that submission, it should, I mean, you should have that information available to you. It will always be Bill landing zone and then your name and then that ID. I understand what you're saying though, why that could be a little bit finer tuned for submitting. That's something that I'm actually going to look into, I think.
Okay, sounds good. Thanks.
So I think by now my data should have finished moving over. So now that that's done, I've submitted my data and my metadata. I'm ready to go ahead and let the Brain Image Library know that I'm ready to make my data public. So we've done that. We've submitted our metadata. Now it's time for the data publication. What you'll do here is you'll send a request for a data validation and a publication request. That will let us know that it's time to run the necessary checks and balances on your data and generate a public link for you. You can do this on the submission portal. There's an option under submit and then a validation request. So if I scroll up, I have this submit validation publish request. And I'm going to scroll down to that one I just created. And you can check off multiple if you're submitting multiple projects and you want, or submitting multiple submissions and you want multiple submissions made public, you can select multiple here. I'm just going to select the one we just did, and then I'm going to submit a validation request. And what this does is it will send direct notice to us that that data is ready to be made public. So once that's communicated from you, what happens is the data will go through a validation process. Essentially, our validation process checks for general things like empty files. It checks to make sure that there's no odd characters or characters that would trip up a file system. And the images themselves get put through bioformats and ensures that they aren't A, corrupted, or B, just black images, things like that. The validation process does not reject, it does not reject based on quality, it rejects on usability. So if the data itself is actually usable. The validation process itself can take time depending on how many files and how large the files are in your submission. Generally, it's rather quick, but there have been situations where it's taken quite a bit of time too. So I've just done that here, and folks can follow along if they want to too, to send that request. Remember, it's this submit publish request, checking off your submission and then hitting that button at the bottom here. And at that point, you're finished, hopefully, right? If the data passes validation, it will be made public. But if it doesn't pass validation, we will reach out directly to you with the best explanation we can give for why it didn't. So now that the data has been made public, you can access the data via the download.brainimaglibrary.org, which is a page you can get to via your browser. Oops. So if you want to find the data that you submitted, this is broken up by the first two characters of the submission ID that was generated, and then the second two characters. And this folder right here might not have something in it, but that's okay. If there was a situation where you created a submission and then it began with 205D, when it becomes public, it'll be located in this directory. You can also access that data within Bill by going to bill/build data, and that's broken up in the same way. So that data can be accessible and usable on our system as well. Additionally, and this will be gone more in depth tomorrow, there's a data search tool we have available. It's located at brainimaglibrary.org/search. This gives a little bit of a nicer view of the submitted data. Oops. Submit.brainimaglibrary.org/search, my apologies. And this is a queryable interface. If you're looking for a specific type of data or your data of your own, you can search from here. There's links to the data itself as well. So if I wanted to view this data here, I could just click the download page and then I would get sent directly to that page we showed previously. So I also want to talk about the PI dashboard that we have on the submission portal. The PI dashboard serves for a PI or a data manager to have a better view of all of the data that's being submitted for the projects that they're associated with. So if I'm a PI and I have 2 different grants that are submitting to Bill, I would be able to see everything that my data submitters have sent so far and the statuses and where they are and things like that. If you give me one moment, I can show that interface. Okay. So as you can see, the interface itself looks a little different. It has more options on the front here for project management, viewing the submissions for all the projects, and the management of the users that can submit for your project. If I wanted to add my project here as a PI, I can do that by clicking manage projects and then create new project, including the information of my project name and if it's part of the BRAIN initiative, as well as the funding information, the grant number associated. If I have multiple projects my folks are submitting for, I can see everything that they've submitted here, as well as information about the data itself, where it's at, if it's been successfully moved over to a public link, or if it's still lingering for whatever reason. And you can manage the users available for your projects as well. In the future, there is a plan to give an option to click and download all of this information, all the metadata and the statuses of the data for your project. Similarly to if you're folks who have submitted to us in previous quarters, sort of a receipt that we send at the end of each quarter. We plan on having that available to users to get on demand whenever they like. So as I said, yeah, it gives a more detailed snapshot of your data by project. And better management for keeping up with exactly what you need to do each quarter. This is just giving some screenshots of that more detailed view. And as I said, there's that, that's our future feature, is this is a self-service data report, which will allow you to get your receipts anytime you like. So that's all I have for the hands-on portions of the submission part. If there's been any questions or issues, please let me know. I'm happy to answer.
Hi, this is Martin. I have two quick questions. One is, if you manage projects and your Brain Initiative funded, but not part of the Cell Census network, I guess you would select no in the first option. Then what do you add at funding? Is it NIH or is it the grant number?
It would be the grant number.
Okay, it will be the grant number.
Yes.
And then I have another question. How do you handle embargoes? Or you don't? Or
Currently, at the moment, we currently handle them by just being let known of a data embargo. There is, we're currently in the works to have some write-ups and procedures for embargoes. We don't have a whole lot of them at the moment, but so it hasn't been super pressing, but it is something that we want to make a bit more intuitive and not just, back and forth emails, things like that.
Okay, thanks.
Like I said, this slideshow will be available to everyone, as well as recordings of the workshop. So if you got tripped up anywhere and you're afraid to speak up or something like that, just let me, you'll be able to take a look at that stuff at a later time, or contact me directly on Slack. I'm happy to help always. But if there's any other questions, so yeah, I got a question real quick about where is the PI dashboard. Sorry, I should have mentioned this. So if you want access to the PI dashboard, you'll have to let us know via the hotline. And it is, you can use it if you're a PI, of course, a program officer, or even a data manager. You'll just have to let us know what projects you would need the PI dashboard for, and we can set that all up for you. If there's no other questions, I would like to I think we're going to take a quick break after this. Is that right, guys?
Yes, we'll take a brief, I don't know, we have it on the schedule for 10 or 15 minutes. So check the schedule here real quick.
Okay. So, hi, my name is Ivan, and I'm going to be talking about some of the computational resources that we have available on Bill for data submitters and for people wanting to use our data sets directly in our infrastructure. For the next 40 minutes, I'm just going to give you a very brief introduction on these resources. Tomorrow, Yana and I will be showing you some exercises and some guided examples that you can do with us that will go deeper into some of these resources that we have available for you. Just a reminder, besides the two Zoom links that you found in the document that you can see on your screen, we also have the Slack workspace. I prefer that you ask questions about infrastructure on Slack, just because if you ask those questions on Slack, they will remain in the chat, whether the questions that you ask here on Zoom, they go away after we end the session. So it would be better if you ask questions over Slack. Both Yana, Luke, and Mariah will be monitoring Slack, and I will be monitoring myself for any questions. If you go to the document that we share with you, the Google Doc, and you go to the agenda, you will see that in my section here, there's a link. And if you click the link, it will open my presentation for today. In case you wanna see it later, or you wanna see what I'm what I'm saying, but this presentation does have some links in them. So just to let you know that you have that resource available to you. Okay, so like I said, I'm going to be talking about some of the computational resources that we have available on Bill. But before we begin, I wanted to set some reminders. If you have any questions regarding any of the resources that we have available or in the workshop, you can send an email to the Bill support help desk, and the email is there on the screen. The link to the workshop VM is the link that you see on the screen as well. Sometimes we don't share these in the documentation for security purposes. So this slide is sort of like giving you the addresses that you need to use to connect to some of our resources, as well as the on-demand link. The location for the data, which is also accessible over HTTPS, and the link to the agenda and to the documentation. The link to the documentation is a little bit different because the documentation that we have created for you is not documentation just for this workshop. It's a documentation for all of the resources at Bill, including more detailed information about the things that we talk about today and that we'll be talking about tomorrow. Okay. For those of you who have never used computational resources before to connect to an HP system like ours, I would suggest that you start by reading this paper that I'm sharing with you. It's a very good reference to start reading before connected to some of our resources, and I will give you a rough idea of what most HPC systems like Bridges 2 or NeoCortex, Bill, or some other data centers across the United States expect from you. It's a very good read. So what is it that you're going to have available during this workshop? Well, Luke already talked about the workshop VM, so you did your previous exercise. That will be available during the workshop, and it will be available to you after for some time. We have 2 8 memory large nodes, normally referred to as L nodes, and they're available to you through a partition called compute. That means that you can submit your tasks, your jobs to that partition. We'll talk about this in a little bit more detail. You can connect to the workshop VM using SSH or X2Go. I will go briefly on how to do that since Luke already talked about this in the morning. You can also access the large memory compute nodes through the VM. So you can submit jobs from the workshop VM as well. And then the new thing that we're sort of talking about today, that if you participated in previous workshops, we didn't, is that now you have access to Jupyter Lab and Jupyter Notebook via open on demand. So this is going to be really quick, but because Luke already discussed it this morning, but you can connect to the workshop VM using X2Go. This is a documentation with screenshots, so it'll kind of like guide you where to go. However, keep in mind that the presentation of this morning is also being recorded and made available to you. But if you click on the full documentation that is on the agenda, you can find step-by-step on how to connect to the workshop VM using X2Go. Again, you can also connect using via terminal. What Luke did this morning was use the terminal within X2Go just to connect to the same machine. But if you run using terminal on your laptop or your desktop, we have an open-facing address that you can use to connect from the outside world. In this case, it's workshop.brainimageslibrary.org. So as long as you have a terminal or terminal emulator on your machine, you should be able to connect to those resources. And if you do, you should be able to perform the same tasks that you did today on terminal when connected via X2Go. Now, if you're not familiar on how to connect using SSH, know that most operating systems, they have either a terminal built in, or it's really easy to download an application to do so. Users of Mac and Linux, they'll probably know They can use terminal or Xterm or some other, like there's others like iTerm2, which are widely popular. For Windows, there should be SSH clients for, I think, PowerShell, but most people like to use PuTTY, which is open source and available as well. The link is there here on the presentation, so you can click and go get the software. So up to this moment, talk about different ways on how to connect our resources, right? We talk about X2Go and SSH, and even within X2Go, we can use the terminal within the workshop VM just to connect to other resources. But now we're going to talk about how we make these resources available once you're connected. So the first tool I wanted to talk about is called LMOD, and LMOD is a library that we use to load software into our workspace. So especially in scientific computing, there's sometimes collisions between different versions of software. And to avoid that, we create these sort of modular environments that allow us to only load into memory or load into our workspace the software that we're interested in using at the moment. And LMOD is widely popular, and it's the one that it's used in most HPC centers nowadays. And we have it available not only on Build, but on Bridges and NeoCortex. So LMOD, it can be a complicated tool, but for the purposes of this workshop, I just wanted to introduce you to 3 commands. Module avail will just list all the modules that we have available or made available to you on Build. Like Alex mentioned this morning, this is the method that we use to install software and make it available to you. But when you run module avail and you don't see the software that you need to use, as long as there aren't any licensing restrictions, we should be able to install and make the software available to you. Module load loads the module of interest, and unloads it. So that's pretty straightforward. If you click at the bottom of this particular slide, it will take you to the documentation of LMOD, with more detail. But like I said, we're trying to keep it very straightforward. For example, running module avail on the workshop VM will list this. By the way, the software that it's available to you via LMOD should be consistent across VMs and resources. The meaning that if there's a software available to you through LMOD and you see it on the workshop VM, you should be able to access it on any L node or any of the login VMs or the submit VMs, etc. So this is sort of to keep it consistently across resources. What else? And then the way you load modules, like I said, is very straightforward. In the previous slide, I show you how to list them, and then you load them by name. The main reason why I'm mentioning MATLAB in this particular slide is because MATLAB is made available through you through Build. However, in order to use MATLAB, you need to complete a form, and you'll see at the bottom of the slide, it says click here. It will take you to a form that you have to fill, and then we can grant you access to this particular resource. But the main idea is that as you develop scripts, well, this is for example, this is loading Anaconda 3. Which is our main Python distribution. And you can see that the IPython binary, it's available after loading Anaconda. The main trick is that whenever you're creating scripts for computation, you can list as many modules as you want and load them in the particular order that you care for. There are times that some modules might conflict with each other. But generally speaking, we take care internally of those conflicts. And if 2 modules are unable to work with each other in the same environment, the script will let you know that you cannot load them at the same time. As far as I remember, there are no conflicts of this type at Build at the moment, but be aware that those can happen. And the script might let you know that, you cannot run with maybe loading, an older version of a piece of software with a newer version of another. Okay. So again, we talk about how to connect to the resources, SSH and X2Go. And then once we're inside and connected to those resources, we use LMOD as a package manager to load software into our workspace or a particular environment. Now we're going to be talking about Slurm. So our Slurm is our scheduler. Again, this is consistent across technologies at PSC. It is the same scheduler on Bridges 2 and NeoCortex. And what the scheduler allows us to do is not only submit jobs to the scheduler, but sort of like partition the resources at each of the compute nodes. So we have 8 compute nodes available to you, but if you only need some limited number of resources in one particular node or in any of those nodes, well, Slurm would allow you to select the amount of resources that you need. And then more than one person could potentially connect to the same compute node, and we can run jobs more efficiently. Just like with LMOD, Slurm, it can be complicated at times. I'm putting the link for the full documentation, but these are the only 4 commands that I sort of want you to know that exist for your benefit. The first one is sinfo. So sinfo would list all of the partitions that you have available. In Slurm, we call them partitions. If you use another scheduler like PBS, this is just another name for a queue. At Bill, we only have 1 partition available, and that partition is called compute. You will always be submitting to compute. However, if you're using another resource at PSC like Bridges 2, then the number of partition increases. Also keep in mind that I think Alex mentioned it this morning, there are no charges against using these partitions. However, if you use another resource like NeoCortex or Bridges 2, there is a charge associated with the partition you're using. And by charge, I mean the number of compute hours that you're using when you submit an allocation request to any of these machines. Technically speaking, you can submit as many jobs as you want for as long as you want. We do put some limitation on the maximum number of hours a job can run. At the moment, I think it's 72 hours. So that's the maximum wall time for any running job. However, you can request any other resource. We don't put any limits on the number of cores or the number or the amount of memory that a particular job should be using. However, keep in mind that we ask some reasonable usage of our resources so that more than one person can access them. Now, the second command is SQ, and SQ is just list the number of jobs in the scheduler, in the queue. You can either list the jobs that belong to you, or you can just look at all of the jobs that are running in Bill, so you can get a better idea when your job is going to start or continue. The next one is sbatch, and sbatch is the command that we use to submit a job to the scheduler. In a nutshell, you create a shell script and then you run that shell script through the command. Again, this is a very brief introduction. For example, if you look at this particular shell script that I wrote, all it does is it loads Anaconda using LMOD, like we talked at the beginning. Then it installs a Python package, and then it runs that package by just printing hello world. And once we do this, we can submit that script. In this case, like I don't need to specify the partition, but I did for the sake of just showing you, because we only have 1 partition available at Bill. And if it gets accepted, it will be assigned a job ID. And when that job stops, you can see the output file. You will generate an output file and an error file. In this case, there were no errors. And cowsay is just a very dummy tool that prints the cow saying a phrase. So technically speaking, this is a dummy job that worked properly on Bill resources. Now, keep in mind that, of course, you will probably be requesting better resources, and your pipelines are going to be more complicated. And then again, sbatch is a more complicated command. At the bottom of the slide, you can click and it will take you to the full documentation. You can there are many other resources that you can request using SVatch. This is just some examples. You can request more memory, you can request more cores or more nodes, depending on the kind of job that you want to run. However, remember that utilizing the resources properly on the L nodes highly depend on how you build your script, or whether the programming language or computational tool that you're using can exploit more than one compute node, or do multi-core processing, or be able to use threads. So that is something that you need to be very familiar with before you start requesting many resources. Or, I mean, technically you could, but just trying to be efficient about it. And then the last one is scancel, which basically cancels your job. You can cancel them by ID, or you can cancel them by user in case you submitted something that is wrong. For bail, like I said, there are no charges. So if you know you submitted something wrong, I mean, we will lose computation time if your job is producing the wrong results. But keep in mind that if you're using another resource like Bridges 2, and you know that your job that is running in the scheduler is not producing the results that you want, you could potentially still be charged for those compute time. So that's why scancel is very important. Now, if you're familiar with Slurm, and some of you might be, you know that in Slurm we have other binaries like srun and salloc, where you can allocate resources to run jobs interactively. So you can connect to the workshop VM, for example, and then connect to a compute node just for debugging or computation purposes. However, in-house, we have built this script called interact, and this is the script that you're going to be using to connect to the L nodes. Again, tomorrow we're gonna show you an example, but keep in mind that this script is we're going to use to connect to the L nodes or any other nodes. The same thing applies for Bridges 2. And with interact, you can request again the number of cores, the partition, the amount of memory, and then you can start an interactive session as long as resources are available to you. Okay, so up to this moment, are there any questions about how to connect to the workshop VM or to any of the other VMs? Any questions about LMOD or how to use SA no charge?
Yeah, I just wanted to just point out that
I see.
Yvonne had used the term charge, and I don't want people to think that necessarily that's a monetary charge. It's a charge against an allocation of a resource, but there's no money that changes hand or anything.
Yeah. I should have clarified that. So depending on the project, you're going to get an allocation, and an allocation is, in a nutshell, a number of hours that you'll be assigned to that project. And when I mean charge, that means that the number of computing time gets discounted from that initial allocation. Now, like I said, there's no charge in bill, but if you're using Bridges 2, the number of compute hours that you get charged depends on the partition or queue you're using. So keep that in mind if you're going to be using another resource like Bridges 2. And the main reason we keep mentioning Bridges 2 and NeoCortex is because the public data is mounted on these two resources. So if you request an allocation in any of these two, you could potentially, do research or do computation and have direct access to the build data without having to download it into any of those systems. Okay, so that's using wrong computer. Okay, and this is sort of like in a nutshell what we talk about. When Slurm, Interact, and LMOD. Now I want to talk about Open on Demand. And the basic idea about Open on Demand is that suddenly all of these resources that I talked to you about before are available through a graphical interface in a web browser. You should be able to connect to Open on Demand. Like I said, through a web browser. You're going to see two addresses at the bottom of the slide. it hasn't been set up properly, but the first one is the link that you should be using in the future. But for now, at least during this workshop, you should be using the one at the bottom. So you can go to ondemand.build.psc.edu, and you can open that in your browser if you want to. I have it open here. Actually, let's see. And it will take you to like a web page that looks similar to this one, and then you can log in with the same account that you used this morning to connect to the workshop VM, which is your Exit account. That is correct, right, Alex? This is the Exit account?
Or whatever your bill account is.
Yeah, whatever your bill account is, that will connect you to that resource. Okay, and that's what I'm going to be talking about next. So if anyone wants to try, go to ondemand.build.psc.edu and log in with your credentials. Please, as soon as you're done, just raise your hand to let me know that we're cool, and then I can continue. I'll give you a couple of minutes. Ian, do you have a question? Okay, so okay, good. I see some people. Please. Okay, so I'm gonna go there myself and I'm going to log in. And once you log in, it should take you to a page that looks like this, right? And the on-demand portal, it's a portal where you can make applications available to you through the web. We have 4 applications at the moment. We have Jupyter Lab and Jupyter Notebook, which are 2 distinct Jupyter applications. Jupyter Notebook is the first iteration of Jupyter. Jupyter Lab is sort of like the latest version. I'm going to be using Jupyter Lab. A lot of people still use Jupyter Notebooks. Now, I want to say that the difference is in the platform, not in the files that you will be creating. Any notebook created in Jupyter Notebook should be able, in theory, to be open in Jupyter Lab and vice versa, as long as the 2 instances have all the packages available to use. The only main difference that I have found using the 2 is that when you're using Jupyter Notebook, each notebook has its own process associated with it, while when you're running Jupyter Lab, you can have multiple notebooks using the same kernel instance. But that it's more like a comment. It's not that important today. So what are we going to talk about? Okay, so we're going to talk about requesting a Jupyter Lab resource. Okay, so I want you to follow this with me. Mine looks a little bit different because I already have a resource requested, but I want you to click on Jupyter Lab first. And when you do, it will take you to a page that looks like this. And I want you to raise your hand once you get to this point. Raise your hand or thumbs up. Thumbs up is better, I guess. Good. I see thumbs up. Okay, so in what happens in this particular form is that we're not going to be using the Jupyter Lab instance directly on an L node, but rather we have to use Slurm to slice the resources so that each of us can connect at the same time. So this is what we're going to do. In the number of hours, I'm going to change that to 1 because this is just an exercise for today. I'm going to set the number of nodes to 2, and you see that extra Slurm arguments here. I'm going to make it bigger. I want you to select this. And what this is doing is sending extra Slurm arguments to Jupyter Lab saying, hey, I want you to assign 16 cores and 32 gigs of RAM to this particular instance. So number of hours is 1. Number of nodes is 1. And the extra Slurm arguments is this. And once you're done, I want you to click launch at the bottom of the page. Okay, so once you click launch, please do a thumbs up or raise your hand so that I know we're in the same page. Good. Good. Okay, good. Okay, so I'm gonna do that, and you will see a page, or you'll see a blog that looks like this that says, please be patient. This might take a few minutes, a minute at a time, or I honestly don't, it really depends on what resources you're trying to request. Okay, but this was pretty good. This is pretty fast. At some point, it should have changed to this little button that you see here that says connect to Jupyter. If you got that button, please raise your hand. If you haven't, or you see an error message, please let us know with a thumbs down, and maybe Luke or Mariah could help. So what information I have in this particular blog? Well, it's telling me that I got assigned to the L node 3, which is important to a certain degree. It tells me how much time I have running on this instance of the Jupyter Lab, and it tells me to click this button to connect. So this, even if I were not to connect right now, this timer will still be running for 1 hour until it goes down to 0, and then it will kick me out. So it's up to you to use those resources. So I click connect to Jupyter. And you should see a screen like this. And if I'm patient, it should turn into something like this. Has it turned into something like this for everyone? Please raise your hand or thumbs up. Let's see. Okay, I see a couple. Okay, good, good. Okay, so I'm going back to my presentation. You can see in the presentation, I put some screenshots so you can follow later in case you haven't. And I gave you some examples. In this case, I said 8 hours, but that's for tomorrow. So tomorrow we will allocate resources for the whole day. Like I said, you click and launch the resource, then you have to wait. And then if everything goes according to plan, it will change to a button that says connect to Jupyter. And with that, you click and it will open a new browser. By the way, at any given point, you can return to this instance. So for example, if you get logged out, let's say for the sake of argument that I request 24 hours and I have that Jupyter Lab instance running for 24 hours, but I get timeout or disconnected from the portal because I didn't use it for a couple of hours, that instance will still be running in the background. So that kernel, that process associated with that Jupyter Lab will still be running in place. Okay. So So what's sort of like the important thing that I really like about Open On Demand? So if you look at the if you look at this particular screenshot, these are some of the things that we can do on Jupyter On Demand, on demand beyond just like running Jupyter notebooks. And one thing is that we can use terminal. So I'm gonna go here and click terminal, and I'm gonna make it larger so you can see. Sorry, it looks ugly, but to make my browser. So this terminal is the same terminal. It will connect you to the resource that you are using. In this case, in my case, L002. And this is the same thing as SSH into the Workshop VM, or can open terminal from the Workshop VM. All my files are available through this terminal. It's just that in this case, it's using my web browser. I have access to all of my resources like Slurm. I can see that some of you are connected. See this list? This list is telling me who is connected to Open On Demand at the moment. So that's good. I see that all of you are connected. And just like I told you about LMod, I can do module avail, and I can list all of the software available. So now I have another method to connect to the bill infrastructure, not only through SSH or through X2Go, but through a web portal. I find this particular connection pretty useful. I know it sounds kind of like odd, but if you're running, if you just have an iPad and you want to check the status of your work, you know, on an iPad, you cannot run X2Go, or it's very difficult. You have to install a terminal client for an iPad, for iOS or for Android. But now that you can connect to our resources using a web portal, then any browser that's available, any portable OS, will allow you at the very least to connect and check the status of your jobs. And I think that's particularly powerful. Okay, so now I'm going to exit, and by exiting, it should close the terminal, and it takes me back to the main page, which is still my instance that is running for 1 hour. Going back to my presentation, like I said, you can use the terminal. The only thing I want to point out that I think it's a new thing that I don't think we talked about in the previous workshop is, and Alex knows more about this than I do in terms of the hardware, but whenever you create a job, when you start a job on Slurm, and all of you connected to Jupyter Lab are requested a Slurm allocation. For each of you, there is going to be a unique folder called /local. And that folder /local, it's a temporary folder that is associated to this particular job. Once this job ends, that /local disappears. However, writing to /local has better performance than writing to your home directory, because as far as I remember, /local are SSDs, right, Alex?
Yes, NVMe.
Yeah, okay. So you could create your jobs so that if you need faster performance, you can write to /local for faster IO. But then at the end of your job, you have to, if there are any files that you want to recover that were written to /local, you have to copy them or move them back to your home directory or your projects folder. So tomorrow we're going to do that. We're going to show you that example. But I just wanted to point out that that's something new that we're presenting for this particular workshop that we hadn't before. The other thing that you can do is that you can monitor active jobs, so you don't have to SSH or use SQ. If you go to Jupyter Lab, well, actually, no, this is on demand, not Jupyter Lab. If you go to on demand, you can monitor the jobs. And in this case, I'm monitoring my own jobs, not everybody else's jobs. And I can see that I have two instances running, two Jupyter Labs, one that I was using to debug some notebooks myself, and the one that I'm using in this particular presentation. And I can kill them. For example, I could just kill them here and it will delete the job if I needed to. But if you were to submit a job using Slurm through your more traditional pipelines, you can also see that. So for example, I created these dummy jobs that they're called sleep because that's what they do. They sort of sleep and they don't do anything other than just wait. I can see the same jobs on SQ. So again, one of the benefits of using Open On Demand is that suddenly you have access to all the same resources that you had before, but now you can do it through a web browser, and it's more flexible. Once your jobs are done, you're going to see them. They inherit the completed label. So you can also monitor the status of your job if you're submitting a more traditional job. And then the other thing that you can do that I find is particularly useful, you can navigate your home directory. And the navigation of the home directory through the web portal is very useful, especially if you want to download a file or a folder. You can also upload from your local machine into your home directory using On Demand. This is particularly useful for small files. You want to upload an image or you want to upload a text file, but this portal should not be used to upload actual data. That's not a good allocation for these resources. Okay, so basically we talk about LMOD, we talk about Slurm, and we talk about Interact, and now we talk about Open On Demand. And On Demand sort of like glues everything together through a web interface. So now you have all of these resources from the web, making them available on the web browser. That means that now you can use additional hardware to just connect to our resources. You can use a tablet, you can use your phone, you can use those Chromebooks as well. So there's no need to go through that additional layer for X2Go or a terminal emulator if you only want to connect for something quick. And yeah, that's about it. That is my very gentle introduction to the resources. Tomorrow we're going to be doing some examples, and in those examples we're going to be like highlighting everything that I talked to you about today. So we're going to go through some workflows. Mine are going to be more simple because I'm going to be highlighting the services that we have available, but Yana's example is going to be more scientific pipeline oriented. It's going to be an actual exercise that will generate useful results. Does anybody have any questions so far? No? Okay, I think, Alex, that was the end of my presentation. I ended 6 minutes earlier than scheduled.
That's fine. Would be happy to entertain any questions that anyone may have. Otherwise, if you need to drop off, you can join 15 minutes or so late tomorrow, because we will be doing a brief introduction for those few people who will be joining us exclusively tomorrow. And you already have some of that. Already had that information. So, but you're welcome to join on time. Other than that, if anyone has any questions, feel free to unmute yourself and ask them. Okay, so since nobody's asking questions, we'll call it a day. Feel free to post questions that you may have in the Slack channel.